Hey guys, welcome back to One Stop Biology. So today we are going to start off with a new chapter, which is chapter 11 of class 12 NCERT, right? So we already finished in the last chapter microbes which were useful for human welfare, right? So in this chapter we are going to start off with biotechnology. So basically guys, we are switching on to a new unit which is of biotechnology, and this chapter is all about what are the principle and processes of biotechnology, right? So let's start with this. So guys, basically what exactly is biotechnology? Let's see that. So if we just break down the word itself, it is bio plus technology, right? So basically it is a branch of science which involves biology and technology both. So basically the techniques that we use, right? So the techniques used with the living organisms, right? Now by using both of these, we basically get some products or processes around it, which is useful to human being, right? So basically, biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products and processes which are useful to humans. So I'll just write down the definition again, that biotechnology deals with techniques of using live organisms or enzymes from organisms. And what do we produce? We produce products and processes which are useful to humans, right? So basically, in this sense, say making bread or making curd or making wine, all of these are microbe mediated processes, right? So all of this could be said as a form of biotechnology, right? But it is used in a restricted sense. Remember that basically what we are doing is we are kind of in most of the times genetically modify organisms to achieve something on a larger scale, right? But this can go in a negative way as well, right? Like, say, guys, nowadays there are so many claims that corona is a lab-made virus. Now, what has it become? Basically, corona is nothing but a bioweapon in a sense, right? Then, right? So, basically, it's not just that, biotechnology can be used in a positive way, it can be used in a negative way as well. And that is why we restrict the use of biotechnology, right? So basically, what we are doing is, say, for example, we do in vitro fertilization, which leads to test tube baby, right? So basically, synthesizing a gene and using it, developing a DNA vaccine or corrective a defective gene, all is the positive part of biotechnology that we use nowadays, right? So basically it's integration of science, the natural science and organism, cells, and the technology part to it, right? Now let's see what are the principles of biotechnology. So basically there are two core techniques that enabled birth of modern biotechnology. So let's see what are these two core techniques. So the first one is genetic engineering. So basically in genetic engineering, what we do is we alter the chemistry of the genetic material. Now this genetic material can be a DNA or the RNA. So it can be a DNA or RNA. And what we do is we basically alter the chemical composition of the genetic material, and we introduce these into host organisms, and we see that there is a change in phenotype of that host organisms, and basically it also helps in treatment of many diseases, right? So we use genetic engineering in by to treat a lot of diseases as well. So basically this is genetic engineering. Now the second one is bioprocess engineering, right? Now what is bioprocess engineering? So basically guys, what happens is that maintenance of the sterile ambience. What do we mean by sterile ambience? Sterile basically, guys, means that the that particular area is free of any microbe, right? So sterile ambience basically means microbial contamination free, right? And maintenance of this kind of ambience in chemical engineering processes is very much required to enable growth of only desired microbe or eukaryotic cell that we want to, right, in large quantities, say, right? So what happens is that, say, if we are producing a medicine, say an antibiotic, right? So we want an environment, a place which is free of any such bacteria that could hamper the production of antibiotics, right? Or in the case of, say, vaccines. So we do not want any kind of microbes or, viruses which are present in you normally in the system to be there, or else it is going to hamper the production of vaccines, viral vaccines, right? So basically, what we do is we maintain sterile ambience through bioprocess engineering, right? We maintain this through bioprocess engineering. So basically, what we have seen is that there are two core techniques that enable birth of modern biotechnology. One is genetic engineering, and the other one is bioprocess engineering. Now let's get into some details for these two techniques. So guys, what happens is that the technique of genetic engineering, which includes creation of, say, recombinant DNA, or use of gene cloning or gene transfer. So basically, in genetic engineering, what we do is we recombine different DNAs, right? And we use gene cloning and we use transfer of gene into the host, right? So basically, to overcome this limitation, and that allows us to isolate and introduce only one set of desirable genes without introducing the undesirable gene into the target organism. So basically, what we do in genetic engineering is we basically target the undesirable gene, we just remove it, and somehow introduce only the set of desirable genes which are required, right? Now how we do it is that a piece of DNA would not be able to multiply itself in the progeny cells of the organisms, but when it gets integrated into the genome of the recipient, it may multiply and it may be integrated along with the host DNA, the person which is the host, and because that person also would be having the DNA, a DNA of its own, basically, right? So this piece, if that gets integrated with the genome of the host, it might multiply and integrate. Now this is because the alien piece of DNA has become part of the DNA of the host, part of the chromosome of the host. Now it has the ability to replicate as well, right? So basically, in a chromosome, there is a specific DNA sequence. Now, this specific DNA sequence is known as origin of replication. Origin of replication. Now, guys, remember that this origin of replication is responsible for initiating replication. So basically, this origin of replication is responsible for initiating. That means starting replication, right? So thus, an alien DNA is linked with the origin of replication so that it can replicate and multiply itself in the host organism. And this process is known as cloning. So now what does it mean exactly, guys? What is happening here is that, say, for multiplication of any, outside piece of DNA, or we can call it alien piece of DNA in an organism, it needs to be part of a chromosome which has specific sequence. And the sequence is nothing but origin of replication, right? So what is happening here is that an alien DNA is getting linked with the origin of replication itself, so that this alien piece of DNA can replicate and multiply itself in the host organism. And this is the process which is cloning. So basically cloning is what? What is cloning? Cloning is making multiple pieces, right? Making multiple pieces, or say multiple identical copies of any template DNA. So cloning is what? Cloning is basically making multiple copies of any template DNA. Now let's focus on one more thing. So let's see the first instance of construction of an artificial recombinant DNA molecule. Let's see how the artificial recombinant DNA molecule is constructed, right? So basically it emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid, right? So how did it exactly came into picture? What basically we thought of is linking a gene which encodes for antibiotic resistance, and we link this gene with a local plasmid, native plasmid. Now what are plasmid? Basically plasmid are autonomously replicating circular extrachromosomal DNA, right? So basically how recombinant DNA molecule, creation of an artificial recombinant DNA molecule was thought of. It was thought to first what? Link a gene encoding for what? Encoding for antibiotic resistant, right? So encoding a gene for antibiotic resistance and encode, basically link it with a plasmid, or say a native plasmid. Now, what is a plasmid? Plasmid is a circular DNA, right? So it is nothing but circular DNA. Basically, it is present as an extra chromosomal DNA, right? So plasmids are autonomously replicating, replicating circular extra chromosomal DNA. It is autonomously replicating extra chromosomal DNA which is circular. So basically we say it is this. This is a plasmid, right? Now we took plasmid of Salmonella typhimurium, right? Now what happened is basically in 1972 Stanley Cohen and Herbert Boyer, they accomplished this artificial recombinant DNA molecule by isolating the antibiotic resistance gene and cutting out a piece of DNA from the plasmid which was responsible for conferring antibiotic resistance. Now, cutting of DNA at specific location became possible only after we got to know about what? Molecular scissors. Molecular scissors. These are basically nothing but restriction enzymes. Restriction enzymes. So basically, we cut these DNA molecules with the help of restriction enzymes. And the cut piece of DNA was then linked to the plasmid DNA. Now, these plasmid DNA, they act as vectors. They act as vectors, right? And they transfers the piece of DNA which are attached to the plasmid. Basically, these vectors, they transfer the piece of DNA, right? So basically, we know that mosquito acts as an insect vector to transfer the malaria disease. Like, so basically, they transfer the malarial parasite into human body, right? In the same way, a plasmid is used as vector to deliver an alien piece of DNA into the host molecule, right? Now, linking of antibiotic resistance gene with the plasmic vector again was possible with the enzyme. So, the way we cut the DNA into various pieces by restriction enzymes, we join or link these DNA pieces with the plasmid vector by another enzyme which is known as DNA ligase. It is known as DNA ligase. So these basically act on the cut DNA molecule and they join their ends, and this makes a new combination of circular autonomously replicating DNA which is created in vitro, and this DNA is known as recombinant DNA. This DNA is known as recombinant recombinant DNA. So basically say this is a DNA vector. You have this plasmid DNA, and then there is a part which is the part of DNA which is to be transferred, right? They are joined together by DNA ligase, right? And we make a recombinant DNA. Right. Now, when this DNA is transferred into E. coli, which is a bacterium closely related to Salmonella. So we took this plasmid from Salmonella, right? Now, when we transfer this recombinant DNA in E. coli, the E. coli will replicate using its host DNA polymerase enzyme, right? So we know that replication happens by DNA polymerase enzyme, right? So this E. coli will use the new host DNA polymerase enzyme and will multiply the copies of this recombinant DNA, right? And this ability of to multiply copies of antibiotic resistance gene E. coli was known as cloning, right? So basically, this is cloning of what? This is cloning of antibiotic resistance gene in E. coli. Right. So now, guys, with this, we can say that there are three basic steps in genetically modifying an organism. What we do is we identify the DNA with desirable gene. So the first one is that we identify we identify DNA with the gene that we want to replicate. So with the desirable genes. Right? This is the first one. The second one is we introduce the identified gene into the host. So the second one is to introduce. Introduce where the identified gene or identified DNA into the host. And third one is what? To maintain the introduced DNA in the host and transfer that DNA to its progeny. So the third one is to maintain. Maintain what? The introduced DNA in which is present in the host and transfer that DNA. Transfer of DNA to its progeny, right? To transfer this DNA into various generations now. So, guys, with this, we have covered the basics, the definition of biotechnology, and what are the what are the principles of biotechnology. So now in the next video we are going to cover the tools of recombinant DNA technology. So I hope guys you would have understood the basics of biotechnology, what is the use and what are the principles. So do give a like to this video, share it with your friends, and yes, if you have any doubt, please do let me know in the comments of this video. You can also ask your doubts over WhatsApp number is given in the description of this video, guys. And yes, if you're new to the channel, please do subscribe to the channel and press the bell icon to get all the updates. Thanks for watching this video, guys. Take care. Bye bye.